My name is Ming-Juan, and Director of Marketing at uBriGene's. I will be your moderator today. So welcome to the RNA Manufacturing 101 webinar. And today's topic will cover the basics of RNA, its different forms, and their respective manufacturing processes. This webinar is a part of our series on advanced therapies, development, and manufacturing. So if you missed our earlier sessions on CAR-T and lentiviral vector manufacturing, you can find the recordings on our website. Before we dive in, I'd like to quickly go over a few logistics. Many of you probably already familiar. During the webinar, all attendees will be on mute, but we encourage you to ask questions at any time by using the Q&A box in the top right corner of your screen. We will hold a Q&A session after the presentation to address as many of your questions as possible. So now it's my pleasure to introduce today's webinar speaker, Dr. Ken Warrington. Dr. Warrington is the Executive Director of Strategic Business Development and Head of Technical Services at uBriGene's. He has extensive expertise in advanced therapies and deep knowledge across various modalities, including live attenuated viruses, virus-like particles, viral vectors, and gene-modified cell therapies. Dr. Warrington has experience in both academia and industry. He worked as a faculty at the University of Florida in the pediatric division of cellular and molecular therapy. He then took on numerous leadership roles in the cell and gene therapy industry, including CDMOs, regenerative medicine, and gene therapy businesses. Ken, without further ado, I will hand over to you now. Thank you.
Thank you, Ming-Juan. And thank everyone for attending today's webinar. It's my pleasure to introduce you to uBriGene's capabilities, growing list of capabilities in the manufacturing of RNA, and to present these capabilities in the context of our workflows that we've developed for GMP manufacturing of a number of the different forms of RNA. So uBriGene offers a number of platform technologies across all of the advanced therapies currently under active development. But for the sake of today's presentation, I'm going to focus on both a little bit on our plasmid capabilities, because it is a critical template for the production of RNA. And certainly focus will be on our RNA product offerings for the manufacturing. And for this presentation, we'll cover at a high level mRNA, self-amplifying RNA, circRNA, and we'll start with small guide RNAs. Briefly, our facilities are listed here. In addition to our centers of excellence for plasmid production and viral vector production in Jinan and Suzhou, we also have a cell therapy excellence center in Guangzhou. And we're excited that we recently brought online our U.S. facility in Maryland to focus our manufacturing offerings there on cell therapies and both viral vector and RNA technologies that enable the development of gene-modified cell therapies. So we'll be able to offer these RNA manufacturing both in our China facilities as well as in the U.S. This is a wordy slide. It'll be you'll be able to access this through our website if you want to revisit it. I just wanted to cover at a high level the 101 part of RNA forms. We'll start with small guide RNA, obviously an area of exciting developments. These are short RNA molecules that guide the Cas9 enzyme to a specific sequence. They're short. They bind to a complementary DNA sequence on the target gene, and they offer a scaffold sequence that interacts and activates the Cas9 enzyme. The activated Cas9 then can edit the DNA at its target site and very precisely modify genes, both at their codon level as well as their transcriptional level. mRNA is a little certainly well known by everyone. It's the single-stranded RNA that carries genetic information from the DNA, where it directs protein synthesis in the cytoplasm. But as we all know, with the COVID vaccine, it can be used to instruct cells to produce therapeutic or antigenic proteins. And these RNAs can be delivered to cells using a variety of delivery systems, including lipid nanoparticles, and can be used to treat a wide range of diseases. And obviously, our clinical experience with the COVID vaccines is they can be produced very quickly. And to date, they're well tolerated and have a low risk of long-term effects. CircRNA is an interesting novel class in active development. It's a class of RNA molecules that form a circular loop, lacking free 5' and 3' ends. They are generated through alternative splicing, where a pre-mRNA is circularized before being degraded. Naturally, they act as miRNA sponges to sequester and inhibit miRNAs. They also act as a scaffold platform for protein-to-protein interactions. They can modulate gene transcription and translation, and they've actually been identified as being associated with specific diseases, but they can also be used as a therapeutic to be delivered to actually treat a broad range of diseases as well. Finally, self-amplifying RNAs are self-replicating elements within them that allow them to replicate within cells, which gives an advantage in terms of increase in the abundance of the protein that is delivered. They serve as a template to produce multiple copies of this desired protein, and again can be used to treat genetic disorders. And also there's a recent approval in the Asia Pacific. They also can be used for the production of therapeutic proteins, but also the approval was for a self-amplifying COVID vaccine. So uBriGene has developed, really focused all of our efforts on the development of RNA production using IVT. And we offer from gene sequence and design through its synthesis and high efficiency transcription template preparation, which the plasmid is linearized and extracted. We then use in vitro RNA transcription generally. And in the case of RNA, can also modify structures with a defined purification and concentration scheme. And we also have a proprietary lipid nanoparticle that we can also package these RNAs that we'll discuss today in a formulation for its cryopreservation. So jumping right into the workflow, starting with the small guide RNAs, our services begin with the offering of target sequence design and determination, the DNA template design, and then, of course, the generation of both forward and reverse primers. These DNA templates are prepared, purified, and then deployed into our in vitro transcription reactions. The linearized DNA templates are digested away, and we have defined purification of the small guides that allow us to aseptically fill and, of course, release with both QC and stability test offerings. Forgive me, I'm losing my voice a little bit. So just showing, again, we offer high quality and have extensive experience in the production of GMP plasmids. We can turn around templates for DNA in a couple of days, leading to high purity of this template, which is critical in our hands for the yield and quality of the in vitro transcribed small guides. This is a few images showing the types of reaction volumes that we use, the quantity of material that we deploy, and ultimately what we recover following TFF. The panel lower right is showing our purification with our target peak coming as a pretty well-defined target peak, and the panel on the right is showing that we can detect the DNA template's purity by gel electrophoresis. These templates are then used in our small guide IVT. We are able to achieve mass ratios in our reaction as high as 100 to 1, and small guide RNA purities at drug substance over 95%. Again, demonstrating here just the purification with the identification of our target peak through in-process purity testing using HPLC, as well as an image demonstrating the detection of the purity of our small guides following purification. We did a direct comparison of IVT versus chemically synthesized guides in our hands, while both IVT as well as chemically synthesized guides we can achieve high purity. We find in our workflows that the IVT has higher gene editing efficiency than the chemically synthesized small guides, and that's demonstrated on the panels on the right, where we're comparing in red IVT versus the purple chemically synthesized small guides in evaluating the editing efficiency. We have a standard and extensive offering of in-house testing. This is a typical QC release testing package for our small guides. I won't go through each one, but obviously appearance, identity, purity, and sterility are critical to the release of these types of moieties. And we offer these in-house to accelerate our customers' manufacturing and release. Jumping forward into our workflow for mRNA, as well as it's essentially the same as self-amplifying RNA. And in this case, added also the fact that we can add to the workflow the packaging of these RNAs into LNPs. Similar approach, linearized plasmids are used in vitro transcription reactions with the second step of capping. We digest away the DNA template. We have established RNA chromatography purification schemes, the ability to then use TFF to concentrate and buffer exchange, and then package as part of the workflow. Excuse me. These SA or mRNAs and the LNPs, which also have a straightforward and single step type purification where we can also fill and store. Just taking a look at the efficiency of our LNPs to deliver RNA. Here, a GFP RNA is being delivered to primary human T cells. The left panel just showing the image under fluorescence. The lipid nanoparticles were added to activated T cells in the presence of serum-free enhancer A. Excuse me, fluorescent images were then captured 24 hours later and the left image obviously showing the fluorescence, with the right showing these cells under phase contrast. They're also efficient at delivery to hematopoietic stem cells, again showing the same demonstration of expression of GFP following the addition of the enhancer into the tissue culture. And these images, again, are 24 hours post-delivery of the LNP. We've designed our self-amplifying RNA to be polycistronic with the ability to express two proteins, and can demonstrate that both of these proteins can be expressed, again, using our LNPs, where in this case, the same amount of saRNA and mRNA were used, but due to the larger size, the molar concentration of the saRNA is significantly lower compared to our linear RNA, but it is demonstrating that both our linear mRNAs as well as our polycistronic saRNAs have robust expression of the delivered proteins using our LNP. Quality control release testing for mRNA and saRNA are similar, if not identical. Again, I won't go through all the list here, but obviously different assays compared to the guide RNAs would be its capping efficiency, poly-A length, RNA integrity, using a variety of methods that we can offer in-house, including potency assays and the development of cell-based, depending on your targets, and the typical safety testing. Excuse me. Sorry, we got a duplicate. Excuse me. Finally, the workflow for our circular RNA LNPs. Again, the plasmid template is linearized and prepared for the IVT reaction. Certainly different here is that we have a circularization reaction as well, with the subsequent steps being what you've seen before. Removal of the DNA template, in this case, removal of linear RNA, a precipitation and SEC purification step, followed by TFF packaging into the LNP, purification of these LNPs, and fill finish. Our circular RNA design uses the PIE method, permutated intron exon, which is relatively a conventional method, although we have some modifications that I'll describe in the next couple steps that have improved our production yields and purities. Using a conventional PIE method, we compared that and began to optimize our IVT reactions. Here looking at a two-step approach to optimization of the production. Using the conventional strategy, the purity for circular RNA was about 45%. But after optimizing both the IVT reaction and the RNase-R efficiency to remove linear RNA, our purities now exceed over 90%. Comparing similarly, looking at circular RNA versus mRNA. In this case, we have the same amount of RNA LNPs, for both of these RNA forms, and these were added to 293 cells in these image, and fluorescent images again were taken 24, and in this case 48 hours post delivery of the LNP, and you can see that there's a comparative expression in the right panel. Looking over time, the circular RNAs tend to have a little higher stability, so the onset was slower, but increased over time compared to the mRNA. Again, we offer a full suite of release testing for our circRNA offering with additional test methods that include RNA circularization efficiency using HPLC and capillary electrophoresis. They look at the residual RNA SAR, the double-stranded RNA, any residual templates. And again, an area of active interest for us and extensive capabilities in the potency assay development, cell-based assays, and the standard safety release testing. So I'll conclude and then we can move on to questions. I just wanted to conclude with a slide where we actually compared all three of these forms. And we saw what one would expect. The self-amplifying RNAs are about 5-fold higher expression at day 1 compared to an LNP that contains mRNA or circRNA and tends to extend out expression longer than the other forms. The circRNA is about 2-fold higher in this example compared to mRNA. In this case, a luciferase assay, where we're looking at 293 cells post-delivery of the LNPs and harvesting at the various time points. I want to thank you again for your attention. I appreciate you taking time out of your day to join us, and we'll be happy to try to address any questions you may have.
Thank you, Ken, for the wonderful, insightful presentation. Yes, as Ken mentioned that we have time for questions. You can put your questions in the Q&A box in the top right corner of your screen. We already got a few questions. Ken, one is asking about our scale of mRNA production and LNP. So what is the largest scale that we can produce?
Yeah, it's a common question. I mean, to date, we've gone as high as 800 milligrams. I'm not sure that's a cap. That's just been what we've performed to date. So about 1 gram to date, and certainly have the capabilities in our process development team to scale further.
Thank you, Ken. And the next question is still related to production. What is estimate timeline to make those RNA LNP starting from plasmid production to the final LNP fill and finish?
Yeah, also a common question and an important one. So it takes us about 3 months to go from plasmid production to a final LNP RNA fill finish. And so I think we take a lot of pride in the ability to turn these products around very quickly, from generating your initial templates to releasing LNP RNA product to the customer.
Thank you. John asked, is our mRNA productions co-transcriptional capping or a post-IVT enzymatic capping?
I believe it's a good question. I believe it's post, but let us follow up with on that one.
Okay. Sure. I believe, I mean, I can chime in a little bit. I think we can do both. We can do co-transcription, capping and post transcription capping. All right. The next question is asking what grades of plasmid DNA we can offer, in just GMP or also research grade.
Oh, no, we offer high quality research grade through GMP. In fact, we take pride in our preclinical grade high quality since we leverage all of our experience, and I'm talking hundreds of GMP batches have been released. So we can leverage that experience to produce very high quality preclinical. And we're excited to engage with clients at that early stage, of course. So whatever a client needs, we can start from preclinical through GMP.
Thank you, Ken. Yeah, and there's a question is if we can, we will send a copy of the deck. Sure, and we can also send the recording, the webinar recording, to you after the webinar. Okay. There is a question is John asked if we offer enzymatic polydinylation of the mRNA.
I believe we do. Again, it's a little deep for RNA. I don't know if you have insight there, but we can follow up on that one.
Sure. Thank you. We can definitely get back to you, John. There's another question. Is overall the differences between the different forms of the RNA, like mRNA, circRNA, and saRNA, in the term of manufacturing processes? What are the differences? What are the similar processes? And yeah.
Yeah, it's a good question, and hopefully it came through in the presentation. mRNA requires in vitro transcription and often includes modifications for stability. I think that's the key point. Circular RNA really offers a naturally stable structure without the need for these modifications, and it can be produced using a self-splicing intron system. saRNA can be produced similar to mRNA, as was demonstrated in the workflows, but it's designed obviously to self-amplify within the cells, and which can ultimately influence, your scale-up strategies in the manufacturing process, because your dose may be, your requirements in terms of yield may be lower than more traditional mRNA.
Thank you so much, Ken. There is a question regarding the advantages of using circRNA over linear mRNA for therapeutic applications. Can you comment on that, Ken?
Again, it's stability and lower immunogenicity in general, compared to more traditional. It's more stable and less immunogenic, and so those are obviously key properties when it comes to developing these as drugs.
Okay, thank you. That's another question regarding saRNA. How does the self-amplifying nature of saRNA impact manufacturing and delivery?
Yeah, as I mentioned, I'm not sure it really impacts manufacturing. I mean, sRNA can amplify within the cells, saRNA. So you can potentially allow for lower doses and a simpler manufacturing process because you don't need to produce as much. But this, again, I'm not an expert in self-amplification processes, but I think they must be carefully controlled, so that you can ensure that your product meets a consistent quality specification to avoid some type of over amplification. I think in general, those are concerns.
Thank you so much, Ken. If you have more questions, you can still put. We still have maybe 5 to 10 minutes. You can still put in your question in the chat box, in the Q&A box. There's one more question regarding how do you optimize mRNA transcription for therapeutic applications?
Optimize mRNA.
Yeah, like
Well,
mRNA manufacturing.
the issue could generally be stability, right? So folks do use modified nucleotides, Again, you're getting a little deep in the weeds for me as I'm an RNA biologist, but I think that's one way that we can also reach out to the team to get a little deeper dive when it comes to these types of questions. Ming John.
Thank you, Ken. We will get back to the customer. All right. There is one attendee is asking if we provide support for CTD preparation and or IND filing.
Yes, we do. We have a pretty extensive experience in both U.S. and China, and have a very talented regulatory affairs team that can support all needs when it comes to IND approval, et cetera.
All right. Thank you, Ken. If no more questions, then we will conclude the webinar. Thank you, Ken, for a great webinar again. And thank you to everyone for attending today's webinar. We appreciate your time and engagement. If you have any further questions,
Sorry to interrupt. I just saw another one from John. Our
okay.
manufacturing facility, as I mentioned in the initial, an early slide, we have facilities in Jinan, Suzhou, and Guangzhou in China that offer plasmid, viral vector, cell therapy, really across the continuum of advanced therapies. And we, in June, we established and became active in our GMP facility in Germantown, Maryland. And these RNA manufacturing workflows are established there, as well as in our China facilities.
Thank you, Ken. I oversaw that question. Sorry about that, John. Any more questions? Please submit in the Q&A box.
Thanks, Benjuan.
Thank you.
Thanks,
yeah. So as I mentioned earlier, that we host a series of the advanced therapy webinars on a monthly basis. Yeah. Please keep an eye on your inbox for information about the next session. Have a great rest of your day, and we look forward to seeing you at the future webinars. Thank you.